myself go away from the screen. Great, thank you so much. Today I'm gonna talk about the spatial distribution of anatoxins in the Scott River watershed. This is more of like updates from what from some of the ongoing work that Quartz Valley has been doing in the Scott River. And so my name is Andrea. I'm a PhD student in the Blast Check Aquatic Ecology Lab at the University of Nevada, Reno. And I want to start off just with some acknowledgements. This work would not be possible without the help of a lot of people and a lot of collaborators, some who are listed here, and some who are also in this meeting. Laurel, who you just heard from, Sarah Schaefer, who is the Environmental Department Director at Quartz Valley, and then Rosalina and Sydney, who are also presenters later on in this session. This is a project funded by the National Science Foundation, and so I'm just going to start off with a couple of background, more of like a planktonic versus benthic. You'll see. So just to jump right in, starting off with planktonic harmful algal blooms, they're pretty extensive and well researched. There's like an established paradigm that they follow. As Maya mentioned earlier, some of these increased nutrients can stimulate some of these harmful algal blooms that just like in the past have been pretty common in the reservoirs. Now with the dams being removed, that might be shifting a little bit more. So one of the things that our lab is really interested in is understanding the benthic harmful algal blooms because they do behave very differently. So with benthic harmful algal blooms, the first thing is that they're really challenging to observe. So here are just some images of some of the mats that we see. These are not from the Scott River, these are from the South Fork Eel, but you can see how sometimes they can be pretty obvious. And like when they cover large areas, you can see them from above, but they can also be a little bit more cryptic. These are from the Scott River. This is Kidder Creek. This is Laurel collecting some of the samples at Kidder Creek. And the thing that's different about benthic HABs versus planktonic is that they do not follow the same paradigm. Benthic harmful algal blooms tend to occur in low nutrient, clear, and fast flowing water. So these proliferations can actually lead to degradation of water quality, similar to some of the planktonic harmful algal blooms. But the one cyanobacteria that we're interested in is the genus Microcoleus, and it produces this really potent neurotoxin called anatoxin. I think some of you may or may not be familiar with it. It targets the nervous system, and it can lead to pretty severe health effects. So here is a map of the distribution of these anatoxins. This is already an outdated map. This is from 2020. It is currently being updated. But a lot of the stuff that we know for like anatoxin production, Microcoleus proliferation, it all has come out from a lot of it has come out from New Zealand and then also from Northern California, which is great because that's where we're located in the Scott River. And we got involved in this project because there is like constant updates of how potent these anatoxins are and how they've been responsible for some of these dog mortalities. So these are just some examples of like some recent work that has come out about anatoxins. So this is kind of where like it's still very unclear how anatoxins are like fully produced by Microcoleus. It's also not the only anatoxin producing cyanobacteria that can occur, but it is one that proliferates in large quantities. And the one thing that we wanted to start off is like looking at all of the environmental parameters that influence or affect Microcoleus growth and production that have been previously researched and synthesized by some of that work in Northern California and New Zealand. And we wanted to break it down a little bit more. We wanted to look at some of the watershed scale parameters. So among them are the watershed area, the geology, the land cover, and then the mean channel slope. So all of these are parameters that can influence the overall stream, but that are outside of that reach Then we move on to the reach scale, which include river flow, sediment grain size, nutrients, big one, benthic light, temperature, and specific conductance. Again, these have all been synthesized in previous work. But we're also interested in these mat scale environmental parameters that are sometimes harder to quantify and study. And these include total biomass and then the total microbial algal assemblages. So this led us to be trying to understand how these parameters influence each other and how they might influence total anatoxin concentrations. And this leads us to our research questions. Our first one is pretty much just what's happening, right? Like what's going on in the Scott River. Laurel and Sarah at Quartz Valley have been doing some of this work since 2022. So we've been really lucky that we kind of got invited to do some of this stuff, some of this research in the Scott River, because one of the hardest things is sometimes you have microcoleus. Maybe it's not toxic. So we knew that there were toxins present in the Scott River, and we wanted to know just what is the spatial and temporal patterns of Microcoleus mat cover that emerge across the entire river network, especially because the Scott River is draining a heterogeneous landscape. And then the second goes back to kind of what I was talking about, the environmental parameters, which is what are which within stream and landscape factors contribute to some of these patterns. So here's a map of the Scott River. These are our sites. In dark blue are our main stem sites, and in light blue are our tributaries. We had a total of 14 sites. And these are just adding on to some of the sites that Quartz Valley monitored concurrently in 2024. So all of this is from 2024, this last, I guess, now a year ago. And just to give you an idea of what some of these sites look like, and here are some of the main stem sites. So the one above is for the Klamath site. So that's where the mouth of the Scott is meeting the Klamath. And then we have Townsend Gulch. And then here we have some of those tributaries that just kind of highlight some of the differences between the main stem and the tributary sites. So here we have Canyon Creek and we have Etna Creek. So you can see how the main stems are more open and the tributaries are definitely more shaded. So just a quick rundown of the methods that we used. This is essentially, this picture kind of encompasses what we were doing in the Scott River. If you drove by and you saw someone doing that, it was probably us. It's very high tech. We used a bucket with a clear bottom. So we cut out and replaced that opening with a plexiglass so that we could see it, use it as our bathoscope. And then we started looking and collecting at all of these environmental parameters that I mentioned earlier. So starting off was water chemistry. So all of those nutrients, dissolved oxygen, pH, specific conductance, it was mostly done using handheld probes and water quality or water graph samples. Then we did our percent cover and velocity measurements. So again, that's a picture showing how we do our percent cover data. We use a 50 by 50 centimeter quadrat and estimated percent cover throughout the reach, which was 5 times the wetted width. And we also collected velocity to kind of estimate discharge measurements for each of our sites. And then we collected mat samples. So from walking throughout the reach, we started to collect a minimum of 10 and a maximum of 15 mats when present. And these were later distributed for microscopy and anatoxin analysis. And then to just kind of expand our understanding of Microcoleus proliferation and anatoxin production, so we collected samples for, like I mentioned, percent cover, anatoxins, and microscopy. However, we have not fully finished our microscopy analysis, so today I'm just gonna focus on the results from percent cover and anatoxins. And overall, we just kind of hypothesized that the local habitat conditions, so those reach scale level parameters, are more important than the watershed conditions, the watershed scale conditions. All right, so what does it actually look like? So to remind you, the first one is just let's see what's going on in the Scott River. How is Microcoleus and anatoxin distributed? So here's another map. It's also, it's just the spatial distribution of the consistency the like overall distribution. So the bigger dots, I mean that's how many times we saw it. And there was only a few of them that were there. So we sampled for 5 months from June to October. And some of them we were unable to reach because in 2024 there was a fire up that kind of closed down some of our site access. And really they're heavily distributed on the left side of the watershed because it's what was publicly available or publicly accessible. So this just kind of shows you a little bit of what that distribution looks like when we actually found Microcoleus. And the first thing that stood out to me was that it's primarily consistent in the tributaries. In the mainstem sites, there were some like Red Bridge and Black Bridge that had some of the mats, but it was really not as well distributed as in the tributaries or as consistent. And then here's the anatoxin. So this is all still data that we're in the process of fully analyzing. And the takeaway here is that we only found anatoxins at Jones Beach, Blackbridge, and Kidder, and one time at the Klamath site. So that large blue dot that you see in the screen there is from Jones Beach, and it is the one that had consistent high anatoxins. Kidder Creek also had consistently detectable anatoxin concentrations, but at much lower levels. But it was present 4 out of the 5 times that we visited Kidder Creek. And in terms of the temporal patterns, there's a peak in Microcoleus percent cover in August that occurs. We were unable to sample all of our sites in July, so I just kept that off a little bit. And then that percent cover kind of decreases significantly in September and October. However, with the anatoxins, we have that peak in October, so it's preceding that peak percent coverage. Which I also thought was kind of interesting overall. So what does that mean for our second question? So like which within stream and landscape factors are contributing to these patterns that we see like spatially and temporally? So just as a quick reminder, these are some of the things that we were interested in looking at. And out of all the data that we collected, there was not any strong correlation for any of the watershed and reef scale parameters. But we are still looking into the microscopy, which might illuminate some of the patterns that occur at the mat scale. So like what's actually producing these anatoxins, especially at Jones Beach, because it had the most, the Microcoleus that was the least that looked like Microcoleus macroscopically, but sometimes in the microscopy, we see something different. So just to give you a quick overview of what the microscopy actually looks like, this is not my area of expertise. Rosalina and Sydney can, might go more into this, but I just wanted to kind of highlight the differences between some of the mainstem and the tributary sites. So in the mainstem side, which is the one on the left here, is Jones Beach, which is heavily dominated by diatoms, but there are some other species that are there that might be producing anatoxins. Again, that's the one that had the highest anatoxins. And then we have our Kidder Creek tributary, which is highly dominated by Microcoleus, but had relatively low toxins. And overall, what this looks like with some of the longer term data collection that Quartz Valley has been doing in the Scott River is that the mainstem sites, even though the percent cover was higher, or the percent cover was lower in the tributaries, the mainstem site had the highest anatoxins. And some of these patterns occur, like we can see here that consistently Jones Beach and Kidder Creek have detectable anatoxins. So that's really like the biggest thing. But what I wanted to show with this graph here is that in 2024, the toxin levels were relatively low compared to 2022 and 2023. So I think it is still important for us to continue, studying or like having these longer term data sets So I'm just going to move quickly because I realize I'm running over time. So our results include that the percent cover was highest in the tributaries in August, and however, anatoxins was highest in the mainstem side Jones Beach in October. And then going back to our hypothesis, really there's still a big question mark. And so how can microscopy help us answer this? So while it's still important, we find that we are struggling to find correlations with just observational studies. I think it's important that we have some more studies that look more at like lab experiments or like in-field experiments that can help us piece out a little more of these mechanisms that are producing high anatoxins. And in terms of monitoring recommendations, this is really, I think, something that Quartz Valley is doing a really good job on is like maintaining this multi-year sampling. That's really needed to understand how this anatoxin is being produced, especially when there's no clear patterns of what's triggering the production. And then the other thing is there are two sites that are consistently the ones that have detectable and high anatoxins, which are Jones Beach and Kidder Creek. So those two sites, it would probably be a good idea to like continue to monitor those. And those are two big recreation areas. The swimming holes there are really popular, so that's another like health consideration as well. And with that, thank you so much. We have time. I'll take questions, and if not, I'll be here.
Thanks, Trey. Yes, we have plenty of time for questions.